All right, so chromosomes, how many we got?
23.
23. 23. And where do you find chromosomes?
Everywhere except for the mature red blood cells.
Yeah. Any place else you don't find them all? I don't think I spelled that right. Is it an A? Is it gametes? Gametes.
ETES, I think.
Is it ETES?
I think so.
And they don't have them all because of why?
This is the only reason male, female.
Yeah, they only have got half of them. You only want half, right? It would be pretty bad if they had all of them, right? Because then the next person would have 46 and then 92, and it would get out of control pretty fast. Are they big enough to be photographed? Yep. And a gene. How about a gene? What's that?
Tell us the specific sequence of proteins.
The chromosome or the DNA has mostly gibberish, but the 23 portions that are going to be chromosomes are divided down into smaller portions of genes, which actually make proteins. So these are just the less than 2% of the actual base pairs that code for any particular protein. And so what's a genes, and how many of these genes do we have in the chromosomes ish?
30 to 40,000.
Yeah, you know what? I actually just changed the notes, and it literally just recently came out to be 19. It keeps dropping. They thought it was 2 million, then they thought it was 100,000, then they thought it was 50,000, then 30 to 40. Now we're down all the way to 19,000.
What's it gonna end up being?
I don't know. Can't be much smaller than 19,000 because although I do say that, and then we'll talk about it later. How do the DNA and RNA bases differ?
DNA has thymine and RNA has uracil.
Yep. So there's a T versus a U, and deoxyribose versus ribose. So sugar missing an oxygen on carbon number 2. Okay, there's the answer to this one. Phosphoester? What's a phosphoester? There's an ester, right? How's the phosphoester differ? What's different about it?
Phosphorus on the worker.
Has a phosphorus where?
Off the worker. Or instead of worker.
The carbonyl carbon's been replaced by a phosphorus carbonyl, or a phosphorus double bonded oxygen. And why are these guys important in metabolism? That bouncing back and forth between ATP and ADP, that's a phosphoester bond. So the base pair itself for adenine turns into ATP when I put 3 phosphate groups onto it. Draw and name one of the 4 DNA nucleotides. Which other nucleotide does it pair with? All right, so Mm. I'm just gonna do this. What I asked, nucleotide? What the? What do we mean by this 5 prime? What does that refer to?
Fifth carbon.
What about the carbon?
Fifth carbon.
Is it fifth carbon? Or is it?
Yeah, a little more specific.
Since the end was the fifth carbon open.
And end to a single strand. So when I've got the strands, one end will be termed the 5 prime and the other term will be the 3 prime end, and that's the carbons on the sugar molecule that extend the backbone of the DNA molecule between the phosphorus and the sugar. So there's a phosphorus that's connected to the 5 prime and a phosphorus connected to the 3 prime, and so phosphorus, sugar, and so one of them, the top is the 5 prime carbon, and the other one, the bottom, is the 3 prime carbon. So it just gives you the orientation of the strand. And how are these guys held together?
Sugar phosphate backbone?
Yep. No. That's here.
Could we also call that a phosphoester?
Sure. Holding them together are the hydrogen bonds between the adjoining A, T's, and C's and G's.
So it's not the end like this bond?